Okay, so in section 4, we're going to look at genetic engineering. So how are the scientists able to uh, put genes of one organism into another? First off, entire organisms can be cloned. A clone is a genetically identical copy of a gene or an organism. This sometimes happen in nature. So bacteria clone themselves. Some plants do that, so you can grow an identical plant from the root of some of them, and some simple animals can do budding or regeneration. So those are steps or things that can be done naturally. So that right there is showing you examples of simple animals that are doing that. Mammals can be cloned through a process called nuclear transfer, and the steps in that process is first you're going to take an egg and you're going to remove the nucleus from it. Once you have removed the nucleus from that egg, you're going to take the nucleus of whatever organism you're wanting to clone, take it from one of its cells, and put it into that empty egg. Then you're going to end up shocking that egg, and that will start the process of cell division occurring. So doing cloning has benefits. You could do this to clone organs for being able to transplant them into humans. They could be used to save endangered species. There are concerns. There is low success rate. Clones are imperfect, usually less healthy than the original animal. Uh, you end up with decreased biodiversity because you're sticking with only certain traits and things like that. The So you aren't getting what would naturally happen out there in the real world. Now, new genes can be added to an organism. This is the genetic engineering. It's going to be involves changing an organism's DNA to give it new traits. Genetic engineering is based on the use of recombinant DNA. So recombinant DNA means you're combining DNA from two different sources. That's what recombinant DNA means. So recombinant DNA contains genes of more than one organism. So right there in that picture, you can see there's bacteria DNA and you've got foreign DNA of some other organism. And the bacteria will read that and say, oh, I need to make this or I need to make this. So bacterial plasmids are often used to make recombinant DNA. Plasmids are loops of DNA in bacteria. The restriction enzymes cut plasmid and foreign DNA. And foreign genes are inserted into the plasmid. So we will be doing a lab with this where we end up taking and you put you're going to use a bacteria that you're going to want to take the plasmid from it, and you're going to put you're only going to want to use one restriction enzyme to cut it, and you will use one restriction enzyme that will cut the human DNA above and below the gene that you're trying to insert. So when you cut it, it'll be able to match up precisely how you want. The sticky ends will match, and then you'll be able to use DNA ligase to put it all together. So genetic engineering, a transgenic organism is an organism with one or more genes from another organism put into its genome. So you can see right there the process that I described of doing this would be how Like we will take and put the gene for human insulin into bacteria, so the bacteria then would be a transgenic organism. The bacteria would not have any use for the human gene of insulin. It will make insulin and it will just get rid of it as a waste product. So transgenic bacteria often can be used to produce human proteins, like I was saying. So genes are inserted into the plasma or into the plasmid, and the plasmid is then inserted into the bacteria. And the bacteria are going to express the gene. We use what are known as genetic markers to make sure that the gene that we're trying to get in there actually gets put in. Some genetic markers would be like antibiotic resistance, and what they would do is they would give the bacteria antibiotics after they would want to check to see did we get the gene in, and if they did, then you wouldn't the bacteria wouldn't die. If it didn't get in there, then the bacteria would die. Another one would be the glow-in-the-dark gene of the jellyfish. You would end up inserting that, and you'd be able to shine a black light and see does the bacteria glow, and if it does, then you know that it got in there. So transgenic plants are common in agriculture. Transgenic bacteria infect a plant, and the plant express that foreign gene. So many crops are now genetically modified or GM crops. So that is something that you've probably heard of, and we will be talking about in class. Are genetically modified crops good or bad, or what's the viewpoint on it? Transgenic animals are used to study disease and gene function, so that's something that's beneficial. Transgenic mice are used to study the development of a disease. So, for example, you can have mice have Alzheimer's and be able to check and see can you cure different issues with it. Gene knockout mice are going to be used to study gene function. So that's what you're seeing down there. You can basically stop a certain gene and see what was that gene supposed to be doing. So scientists have concerns about some uses of genetic engineering, possibly the long-term effects of eating GM foods, possible effects of GM plants on an ecosystem and the biodiversity are some of the common concerns for it. So that is the end of the notes for section 4.